Hey everyone, this is Mr. Baker. We are going to talk today about our third section, cell growth and reproduction. The growth part is for the cell cycle here, right? I would like you to draw this diagram in your notes. So if you haven't yet, hit pause, draw it. You should recognize the cells with chromosomes, little squiggly lines or X's inside them. This is the pattern that cells follow as they grow, right? We will start talking about this in the G1 phase, which is the gap one. The cell grows physically larger, copies all of its organelles. It does not copy chromosomes yet, but all the molecular building blocks are put in place and everything's getting ready to go. S is the synthesis cycle, right? It makes a copy of DNA. This is DNA reproduction. We've already talked about this, right? It also duplicates centrosomes, which help separate DNA later. G2 is gap 2. It grows more, it makes some proteins and organelles, and organizes its contents in preparation for division. It's kind of a double check phase. It makes sure everything is ready to go for division, which is called mitosis. If the cell is not going to reproduce or split into two, right, it could be on its way to death. Programmed cell death is apoptosis, or it could be going stagnant, where it has reached a point of maturity in that cell's life and it's not going to do much except for what it's already doing. That's the G0 phase in the bottom right-hand corner that you see where it says cell cycle arrest. That just means it's not going to do any more growth. It's not going to be doing any more replication. It's just going to be doing whatever job it is doing right now. The skin cells, not at the very top layer, but in the layers just below those, are a great example of cells at cell cycle arrest. The replication part is called mitosis. We're going to talk about each of these steps in the next slides, but I want you to know that mitosis makes exact genetic copies, right? 1-to-1 copies. That's the goal. Sometimes there are mutations that happen, sometimes there are errors that occur, but the goal here is to make exact copies of the cell as it is in its kind of mature or adult phase. The first step is interphase, right? And when I say first, I mean kind of, because it's actually all that part that's not mitosis. If we go back on our diagram here, interphase is G1, S, and G2. So we already talked about interphase, but if we're talking about the cell cycle and mitosis, interphase is kind of understood to be the general step, right? The first real part of mitosis is prophase, right? Prophase is where the centrosomes move to the opposite sides of the cell. I told you that those were made during S. They're going to help with DNA splitting. But the DNA also condenses. So instead of being those long strands, they are made into chromosomes, wrapped around histones, supercoiling, all that stuff. And as they do that, right, because we've already replicated our DNA, a centromere is going to join what we call two sister chromatids. Those are the exact copies of the DNA, right? You as a human have 23 different chromosomes, but you have two copies of each, right? Each of those 46 chromosomes is actually doubled here. All right, so we get two copies of each chromosome. All right, and then our nucleus goes away. So the nuclear envelope disintegrates because everything's going to be moving around. Next is metaphase, where those chromosomes are actually pulled together towards the center through something what we call an equatorial plane or a metaphase plate. It's not a real recognizable location so much as it is the middle, right? The centrosomes at the poles are going to pull on their spindles. Those are those kind of yellow line looking things that connect the centrosomes to the centromeres, right? Until they are taut, and that lines everything up in the middle. Anaphase is when the centromeres are actually broken down and the tension on the spindle fibers actually pulls the chromosomes physically apart towards opposite ends of the cell, towards the poles. What we were calling sister chromatids are now split, and we call them daughter chromosomes. I'm sorry that there's a lot of C words here. I didn't get to name anything. Telophase is the last real step of mitosis, where pretty much we're undoing all the things that happened during prophase. The nuclear envelope is going to form back around each of the groups of daughter chromosomes, making daughter nuclei. The chromosomes are going to uncoil as those nucleoli appear, right? And the mitotic spindles are actually going to break down back into parts that the cell could use for other things. And cytokinesis starts. Cytokinesis is a process inside mitosis, but it's not its own step. It actually starts at the end of anaphase. It will go through telophase and will complete after telophase ends. And depending on if you're an animal cell or a plant cell, this looks a little different. Either way, the cell membrane itself begins to pinch and make a cleavage furrow. You'll see that in the next slides as we've got some more images coming up. In plant cells, there are some vesicles that are going to align, and they're going to form a cell plate, which begins making a cell wall. Animal cells, because they don't have a cell wall to make, that ring that is pinching the cytoplasm is actually going to keep pinching until it divides the cell completely in half, making half on one side of the new cellular membrane, one on the other half of the cellular membrane, and we get two genetically identical daughter cells. I'm not going to ask you any questions this video. I do want this version of the diagram in your notes. Honors class, we are adding prometaphase to what we just talked about. Regular biology class, you can actually skip that one in the top right that's labeled prometaphase. Here's a simplified version. It kind of helps for studying, but it's not the one I want you to draw in your notes. We will continue to talk about mitosis over the next couple of days. I will see you in class.